really just taking a deeper look at what reproduction really means. So last time we went over meiosis and the mechanics of how we can create two gametes, two special cells with half as much genetic information as a parent cell, so that we can combine two gametes and make a new member of the same species. All of that was worked out much later than the stuff we're going to talk about today. Today we're going to talk about the guy named Gregor Mendel, who's often called the father of modern genetics, even though he didn't know what he had done. He never knew that anyone was going to care about his work. But he did the experiments that were rediscovered after his death that allowed science to explain how dominant alleles mask recessive alleles and gave us two of the laws that exist in biology, two very important laws that are going to help us understand reproduction of organisms. So your objectives here are to explain the significance of Mendel's experiments on the study of genetics. I want you to be able to summarize the law of segregation and the other law, the law of independent assortment. We'll go over those. And something else that you're going to be able to do here, but this will probably be clearer once we get to the chalk talk part, is to be able to use something called a Punnett square to predict possible offspring if you know their genotype. So let's start with a vocab. Segregate. You probably heard the word segregate before. What you need to understand in genetics, every trait has two genes that control its expression. So if we're talking about eye color, hair color, height, width, shape, any trait we can has two genes. One gene came from mom and one gene came from dad. They're the same gene, they're just two different copies of that gene. Those two different copies of the gene have to separate, and that's what segregate means, to separate. And we know that from meiosis, when the chromatids were separating from one another in the formation of gametes. So each gene that has two copies of itself must separate from one another during meiosis, or gamete formation. So if we have two genes, that means we are two copies of the same gene for the same trait. We need some language to describe what specifically we're talking about. So we're going to call all those different forms of a gene alleles. So you got an allele for hair color from your mom. You got an allele for hair color from your dad. They're both genes that control hair color, but they're different forms. And those forms are going to have two possible types so far. We'll get into some other details later. And those forms are dominant and recessive. If it's a dominant allele, it covers up the presence of a recessive allele. And those covering up recessive, dominant traits are going to have ways they look. And those ways that things look are called phenotypes. So you have a gene for the color of your hair. You actually have two copies of that gene for the color of your hair. You have one you got from your mom and one you got from your dad. One of those alleles might be dominant and one might be recessive, but the way those two genes interact with each other, the way those two alleles interact with each other, are going to give you hair color. And your hair color is your phenotype. So a phenotype is always going to be an adjective that describes a trait. Tall, short, green, yellow. So we all have these genes. They occur in pairs. We got one allele from mom and one allele from dad. So we're going to call the actual alleles, the actual two genes we have, a genotype. And that means you can either have two that are exactly the same, which would be homozygous, or two that are different. If you got one allele from your dad for brown hair and an allele from your mom for blonde hair, you would be heterozygous for your hair color allele. If you had two blonde hair genes, one from mom and one from dad, you'd be homozygous for your hair genotype. And a hybrid is very similar to heterozygous. It's a cross between two homozygous individuals. Those two alleles have to separate during meiosis. There's one on each of your chromosomes. We said you had one chromosome from your mom, one chromosome from your dad. And that gives us the law of segregation. And the fact that crossing over happens gives us the law of independent assortment. So the genes for your hair color don't care where the genes for your height go. If you inherit a hair color allele from dad, you can still inherit the height gene from your mom. And one of the reasons I like this section a lot is this guy, Gregor Mendel, father of modern genetics, one of the most mentioned people in all of molecular biology probably, is not that much different than you or I. He was born in 1822 in a farming community in Czechoslovakia, or what was Czechoslovakia. 1822 was quite a while ago. Ulysses S. Grant was born in 1822. Rutherford Hayes, Louis Pasteur. Even this state, the state of Maine, was only 2 years old at the time. Mendel spent his childhood working on the family farm, but his father didn't think he was strong enough to be a farmer, so he made sure that Mendel went to school, and Mendel went to school to become a teacher. The language of the place he was living in, has grown up in, was German. The place where he was sent to teach was a different language, so there was an accent, there was trouble making connections with students, and he was fired from his teaching job. He then went on to teach college, but he couldn't pass the exams, so he couldn't get credentialed to be a college teacher. So up until this point, he had not failed in life, but he had not made any of his goals. He was a priest working in a monastery, so the monastery gave him a job to do. They put him in charge of the gardens, and he decided to do some scientific experiments. He was very good at math and statistics. He was interested in biology. A lot of people at the time were interested in biology. So from 1856 to 1863, roughly the time of the American Civil War, he grew about 28,000 pea plants. And he published the results of his experiments in a paper called Experiments in Plant Hybrids in 1866. Nothing happened. That paper was hardly ever read. It was very important. It was rediscovered years later. But as far as Mendel knew, nothing ever became of that. It was just one more of those things that didn't work out the way he hoped. In 1868, though, he did get promoted to the head of the monastery. He was a very smart man. He did things to help him. In the process of the stress of running a monastery, though, he became a heavy smoker. There were no warnings on cigarettes at the time. He developed kidney problems, which you can get from smoking. And in 1884, he died. Again, not knowing what he had contributed to modern biology. He had set out to become a practical breeder. He wanted to figure out which plants he should cross to get the best possible offspring. People have been trying that forever. People had good ideas. Everyone believed that if you cross two things, you get something in the middle. If you cross a tall animal with a short animal, you should get a medium-sized animal. Mendel found out when he was doing his experiments that it was very difficult to explain what he saw based on that blending hypothesis. So he turned to mathematics and figured out that it was not really a blending. He used the scientific method. He observed. He looked at pea plants over and over again. He made his hypothesis, and his hypothesis to begin with was blending, that you would get something in between the two extremes. But when he did his experiments, he had to conclude that blending was not the correct hypothesis, that for the things he looked at, and what turns out to be true for most things, is you don't get a blending. You get a dominant or a recessive, or some other interaction we know now. So he revised his hypothesis and started all over again. He started with garden peas, and garden peas were very important for him to choose. Garden peas reproduce sexually. Not all plants reproduce sexually. If you didn't reproduce sexually, you didn't have two distinct sex cells, two gametes, he never would have figured out what he was trying to figure out. And in addition to that, in garden peas, he could control the fertilization. In garden peas, he knew to take the male part of the plant and sprinkle it on the female part if he wanted to cross the plants. And he knew that pea plants are self-crossed or self-fertilized in most cases. He also had the ability, by using pea plants, to have a number of true breeding plants. True breeding plants always produce the same phenotype in the offspring. So if you started with true breeding tall plants, you would always get true breeding tall plants. And we know now that that true breeding means they were homozygous. They had the same allele at both the mother and father chromosome. And the other thing that was very important for him to do, to figure out mathematically especially, was he only looked at one trait at a time. His second law deals with when you look at two traits, but to get started, he only looked at one trait at a time. So we do need to go into some more vocabulary terminology. Let's start with phenotypes. Phenotypes are descriptions of a trait. So if we're talking about plant size, it turns out we have two possible phenotypes in pea plants. You can have a tall pea plant or you can have a short pea plant. There are no medium pea plants. That phenotype is determined by the genotype of the adult plant. So the adult plant can either have two copies of capital T, capital T. They can have two of the tall alleles. They can have two of the short alleles, which we're going to call S and S for now. And if that's the case, you are homozygous. You could also have one tall allele and one short allele. In that case, you would be heterozygous. So homozygous means those two alleles are the same. Heterozygous means those two alleles are different. As far as the generations go, we start with the parental generation, and we call that the P1. So the P1 are parents that have two copies of the allele for the gene for height, and they can pass on one of those alleles to the first generation offspring. That first generation offspring is going to be the F1 generation. If you cross two members of the first generation, you get a second generation, and we call that the F2 generation. And we do that in order to figure, Mendel had to do it, to figure out what the genotypes of the plants were. So we're going to start with what is called a monohybrid cross, which means we're going to look at one trait, and we're going to look at parents that are different in that trait. So we could cross a true breeding tall plant with a true breeding short plant. In order to do that, Mendel took the male parts off the plants, dusted the female parts with the pollen where he wanted to, then covered the flower with a bag so that nothing else could cross-pollinate it as a safety measure. So when he did that, his true breeding tall crossed with true breeding short, and if we write it in shorthand, we can have capital T for the true breeding tall. And now we're going to call the short lowercase t, and I'll get into that detail why we do that later on. When you do that, the only possible offspring you can get are one capital T from here, one lowercase t from here. So all the offspring are going to be capital T, lowercase t. They're going to be heterozygous. But they are all going to have the phenotype of being tall. So that doesn't tell you much except that short disappeared in this generation. So the question is, did short really disappear, or is it just hidden? And in order to figure that out, Mendel took these tall pea plants from the first generation and crossed them with one another. So capital T, lowercase t, crossed with capital T, lowercase t. And when he did that, he got 3 tall offspring and 1 short. That told him, number one, short didn't disappear. It's still there, hidden in the first generation. And number 2, it can come back, and it comes back in a very specific ratio, 3 tall and 1 short. That means it's not blending either. He repeated that same experiment with 6 different traits. So he had 7 different traits that he could do monohybrid crosses with. The results were always the same. The first generation, one of those 2 phenotypes disappeared. In the second generation, that phenotype came back in a 3-to-1 ratio to the other phenotype, which led him to the conclusion, as I told you earlier, genes occur in pairs. For every trait, we get a copy of the gene to give it to us from our mom and a copy of the gene to give it to us from our dad. Those 2 factors are going to interact with each other. Mendel had absolutely no idea what those factors were. He didn't know if they were a physical particle. He didn't know where in the cell they were. No one did. But so he just referred to them as particles of inheritance. We now know that those are genes. So for every trait he looked at, there is a gene. And every organism that has that gene for that trait has 2 copies of that gene, or 2 alleles for that gene. And for everything that Mendel looked at, the 2 possible forms were a dominant form and a recessive form. The dominant form was seen in a first generation, and the recessive was not. The recessive was hidden in the first generation. And in the second generation, you got 3 dominant phenotypes for every 1 recessive phenotype. So the law of segregation tells us that those 2 factors, again, he didn't know what they were, that now we know they are alleles. The 2 factors that control a trait separate during the process of gamete formation. So a parent can only pass on one of the 2 copies they have to their offspring. The law of independent assortment, you have to look at a couple of crosses at the same time. So if you were looking at the gene for height and the gene for color, the gene for height doesn't care where the genes for color go. So this is all completely independent assortment. You're not going to pass on an entire maternal chromosome or a paternal chromosome. This gets to that crossing over. And again, this is where we get the randomness from sexual reproduction. Gamete formation is random. Gametes coming back together is random. So if we look at Mendel's monohybrid cross, Mendel took a tall pea plant and a short pea plant and fully expected that when he crossed them, he would get a medium-sized pea plant. When he did that cross, though, he did not get any medium-sized pea plants. Every single one of his offspring was a tall pea plant. So to decide whether or not that shortness had disappeared, whether it was hidden, or if he even made a mistake, he crossed 2 of those together. And when he did, he got a ratio of 3-to-1. And he didn't just get 4 plants. Remember I told you he did 38,000 plants when he checked these over the course of 4 years. So he came up with an idea that there must be 2 things that are controlling each one of those traits. Those 2 things must separate when you form a gamete. And when those gametes come back together, then everyone would have 2 copies. And the way those 2 copies interact with one another is going to determine the phenotype. And if you have the dominant allele, either 1 copy or 2, you're going to have the dominant phenotype. If you have the 2 copies of the recessive allele, you will have the recessive phenotype. And we can use a Punnett square. I'm going to go quickly, but this will be the focus of my chalk talk tomorrow. This is a shorthand method for following the traits. If you know the genotype of the parents, you can use a Punnett square to predict the phenotype of the offspring. And it looks like a box. In this case, we're looking at 1 trait, so it would have 4 possible combinations. This is 1 parent, capital T, capital T. This is the second parent, lowercase t, lowercase t. Those are the parents in the cross, and then we have to figure out what gametes they can make. That parent can make one gamete that has a capital T in it and another gamete that has the other capital T in it. The other parent can make a gamete that will have a lowercase t and a different gamete that will have the other lowercase t in it. Now all we have to do is predict how they come back together. So we take this t into this box, this t into this box, and so on. And we see that all four boxes contain a capital T and a lowercase t. So this is Mendel's first cross. Capital T, true breeding tall, crossed with lowercase t, true breeding short. And all the offspring were tall, but they were hybrids. They were not true breeders. And 3 out of 4 of their offspring would be tall, the fourth one being short. So it's all about probability, kind of like flipping a coin. There's two sides to a coin. There are two possible alleles in a gene, in these genes anyway. For example, you could have the capital R round allele or the lowercase r wrinkled allele. And this is all about randomly distributing traits. So the study of genetics began with Gregor Mendel. His experiments with the garden pea gave insight into the inheritance of traits, a great deal of insight. He developed the law of segregation and the law of independent assortments. And after him, when we figured out what was going on, a guy by the name of Reginald Punnett developed a shorthand method for following monohybrid, even dihybrid crosses are not very hard with Punnett squares. All right, talk to you tomorrow.